Dear viewers, our today's lesson's objective is to show you the definition of allele specific primers. A brief introduction to ARMS PCR. Characteristics of allele specific primers. Designing allele specific primers. And the use of allele specific primers with animations. So, let's begin. Allele-specific primers are the primers that specifically amplify only one of the alleles. They are used in ARMS PCR. Okay, what is ARMS PCR? It stands for Amplification Refractory Mutation System. It is an application of PCR in which DNA is amplified by allele-specific primers. Test DNA is only amplified when the target allele is contained within the sample. ARMS PCR is mostly done to identify a mutation or a polymorphism. It can also be used to identify whether the change in DNA is heterozygous or homozygous. Characteristics of allele-specific primers. Although you can use two or more than two primers in ARMS PCR, two of them are compulsory. A common primer, which could either be forward or reverse, but it must bind to all the alleles. The second primer must be an ARMS primer, which specifically bind to your target allele. If common primer is forward, then ARMS primer must be the reverse and vice versa. Depending on your research objectives, you can also optionally use a third or a fourth primer as well. The common primer is like any other PCR primer. However, ARMS primers have the following additional specific features. First of all, it must follow all the characteristics that we use for designing a common primer. The link to the video is given here. You can watch that video for details. The specific feature of allele-specific primer is that the nucleotide at the 3' end of the primer must be complementary to the nucleotide at the SNP position of the target allele. Now let's use BatchPrimer3 to design allele-specific primers. Step number 1. First, you have to get your desired sequence. Now identify the target nucleotide at the SNP position. For example, in our wild-type allele we have T at the target SNP position, while our mutant allele has C at this position. After you locate and identify the nucleotides at the SNP position of all your alleles, replace them with a single letter that can represent all the nucleotides at the SNP position. This table contains all the letters that can show ambiguity among nucleotides. As we have T in one allele and C in another, therefore we will have to replace them with Y in our sequence, as shown in the table. Okay, now replace TC with Y. Now open the BatchPrimer3. You can search this site on Google. Now click here. Once the site is opened, click here to open the drop-down menu. Then click this option. You can also select this option. Paste your sequence in this blank space. Then click this button, and that's it. Your allele flanking and allele-specific primers are ready. These are the primers we obtained. These are common allele flanking primers. And these are allele-specific primers. We have two kinds of allele-specific primers, wild type and mutant type. Each of these primers have forward and reverse primers. Note, this is not always the case. For some sequences, you may not get one or more of these primers, or you may not get any primers at all. You can use allele-specific and flanking primers for a variety of purposes. The three main uses are the identification of mutant or wild-type allele, identification of a nucleotide at a specific position of a sequence, and identification of homozygosity or heterozygosity. Although there are several methods for SNP identification, in this video I am going to tell you about the use of ARMS PCR. For this PCR you would mostly require 3 primers. 1 common primer, 1 wild-type allele-specific primer, and 1 mutant-type allele-specific primer for each allele. To detect or identify alleles or identify homozygosity heterozygosity you can use either of the following groups of primers. In 2 primers method, depending on your research objectives, you can select one of the following 4 pairs of primers. Number 1. Forward allele specific primer for mutant and common reverse primer that can bind to all the alleles. Number 2. A reverse allele specific primer for mutant and a common forward primer. Number 3. A forward allele specific primer for wild-type allele, and a common reverse primer. Or, reverse allele specific primer for wild-type and common forward primer. In 2 primers method, you saw that we selected only one of the primers from the allele flanking and one from allele-specific primers. There is another method in which you use one from allele-specific primer and both the allele flanking primers. This is called 3 primers method case 1. You can use any of the following groups of 3 primers. Number 1. A forward allele-specific primer for mutant and both the flanking primers. Number 2. A reverse allele-specific primer for mutant and both the flanking primers. Number 3. A forward allele-specific primer for wild-type and both the flanking primers. And number 4. A reverse allele-specific primer for wild and both the flanking primers. In 3 primers method case 1, you used both the flanking primers but only one from specific primers. There is case 2, in which we will use 2 specific primers but only one from the flanking pair. In this method, you can use either of the following 2 groups of 3 primers. Number 1. A forward specific primer for mutant and a forward specific primer for wild, while from the flanking primers only reverse primer will be used. In method number 2, you use reverse specific primers for both the alleles, and only the forward primer from flanking primers. There is another method in which we use all the 4 primers, that is both the allele specific and both the flanking primers. In this method you can apply the following 2 groups of 4 primers. Number 1. A forward primer for mutant, a reverse primer for wild and both the flanking primers. Number 2. A reverse primer for mutant, a forward for wild and both the flanking primers. In 2 primers method, you can use either of the following pairs of primers. This has already been explained in the previous slide in details. Each of the above 4 ways have 2 possibilities. That is, either the target allele is found or not. Here in this video, I am interested in the mutant allele only, therefore I will show the use of forward or reverse allele specific primer for mutant only. The use of forward and reverse specific primers for wild type are exactly the same. Primer pair 1, possibility number 1, that is, mutant allele is present in the sample. First you put your sample in the PCR tube. As we have already supposed that this sample contains the mutant type C allele, so one of the DNA strand will have C and the complementary strand will contain G. Now add the forward allele specific primer for mutant, which will bind at the SNP position during PCR cycles. As you can see, the 3' end of this primer has C, which is complementary to G, so it will amplify the strand. Okay, now add common reverse primer for making the exponential amplification possible. After adding all the required reagents to the PCR tube, run the PCR. During PCR, the target allele will be amplified. Now run the gel. As the amplicon is 300 base pairs, therefore you will get a band at that position on the gel, which shows the presence of the target allele. You can also run positive and negative controls with PCR to make your results more reliable. Now let's come to the possibility number 2. What if you have no mutant allele in the sample? You have the wild-type allele and you are using the forward primer for mutant type. As you can see that the 3' end of the primer is not complementary to the wild-type allele. So the 3 end of the primer will not be able to bind. As a result, you will get no amplification and hence no band on the gel at around 300 base pair. Primer pair 2 and possibility number 1. Mutant allele is present. In this method, you use forward common primer and reverse mutant specific primer. As the primer's 3 end is complementary to the SNP position in the DNA strand, therefore you will get the 200 base pair amplicon which will be detected through the gel after PCR. The next possibility with the same primers is that you have only the wild-type allele in the sample, then the primer's 3 end will not bind to T in the target DNA because G of the primer is not complementary to the T. So no amplicon and no band on the gel. Now let's come to the 3 primers method. This is case 1. The details is already explained in the beginning of the video. You can use any group of the 3 primers. Again, I am only interested in the mutant allele. Therefore, I will use the first 2 groups of primers. Triplet of primers number 1 and possibility number 1. We suppose that our sample contains the mutant allele. First, you take your DNA sample. Then add all the 3 specified primers. As allele specific primers 3 end can bind to the allele target site, therefore you will get the amplicon of 300 base pairs. Because we are using both the flanking primers, we will get a 400 base pair long amplicon as well. You will get both the bands on the gel. Possibility number 2 with the same primers. Suppose you do not have the mutant allele. Then your allele-specific primer for mutant type will not give any amplicon. But you will get the 400 base pair amplicon, because you are using both the flanking primers. So you will get only the common amplicon, but not the specific amplicon. Triplet number 2. Possibility number 1. Mutant allele is present. Take the DNA. Add all the 3 primers. In this case we are using the reverse mutant specific primer. Run the PCR. As the primer is mutant specific and mutant is present in the sample, therefore we will get the amplicon of 200 base pairs. When you run it on the gel, you will see both the common as well as the specific amplicons bands on the gel. In this possibility we have only the wild-type allele in the sample. Using the same group of primers, we will get only the common amplicon, but not the mutant-specific amplicon, and hence on the gel only one band of 400 base pairs will be seen. Now let's come to the case 2 of 3 primers method. It has got 2 methods. In method 1 you use forward common primer while reverse primers for both the alleles. In method 2, you use common reverse and both the forward primers of wild and mutant alleles. Remember, you will have to use separate PCR tubes for allele-specific primers in this method. Mutant-specific primer will be added in one tube while wild-specific primer will be added in another tube. However, if you want to put both of them in a single tube, then you will have to label them so that you can detect them later from their labeling. This method has the following 3 possibilities. Mutant homozygous detected. Wild homozygous detected. or heterozygous detected. In this case, we are using the reverse primers for both the alleles and forward common primer. Here we suppose that our sample is homozygous wild type. Means, our sample contains only the T allele. First, take 2 PCR tubes, one for wild specific primer and one for mutant specific primer. Now label them. Then put your sample DNA in both the tubes. Add the common primer to both the tubes. Add the wild specific primer to the tube, labeled as wild. And add the mutant specific primer to the tube, labeled as mutant. As we have only the wild type allele, therefore the tube that has wild specific primer will amplify the DNA. After the PCR, put the PCR product of both the tubes in separate wells and run the gel. You will get a band at around 200 base pairs in the wild type lane, while there will be no band on the mutant type lane. Now suppose your sample contains only the mutant allele. Means a homozygous mutant is present. Perform the procedure as shown in the last slide. This time the DNA in the mutant PCR tube will be amplified and you will get the result on the gel. In this third case, we suppose that your sample is heterozygous, means both the alleles are present. Put the sample in both the tubes. In wild PCR tube, only the wild type allele will be amplified, because we have added the wild specific primer to it, and it contains both the alleles. The mutant allele present in wild tube will not be amplified. In the mutant PCR tube, only the mutant allele will be amplified. When you run both the PCR products on the gel, you will get bands for both the alleles. Now let's change the primers. Here we are using the forward primers for the alleles while reverse common primer is taken from the flanking primer pair. You put your sample in both the tubes. You added the wild specific primer to one tube and mutant specific primer to the other tube. Common reverse is added to both the tubes. As this is a homozygous wild allele, therefore only wild allele will be amplified and you will get a 300 base pair band on the gel for wild type only. In this case you will only get a 300 base pair for mutant allele only. Here again both of your alleles will be amplified and both the bands can be seen on the gel. Now let's come to the 4 primers method. There are 2 ways of using a group of 4 primers. In both the methods you use both the flanking primers. In method 1, you use forward for wild and reverse for mutant, while in method 2, you use reverse for wild and forward for mutant. Let's come to the wild homozygous case. Here you use only one PCR tube. You put your sample in a single PCR tube. Add both the common primers, forward for wild and reverse for mutant. You will get 2 amplicons, one between the flanking primers, and one from the wild specific primer. Therefore on the gel you will get 2 bands at relevant positions. Now we have mutant homozygous case. Again you add the same primers. But this time you will get one amplicon of common primers and one of the mutant allele. On the gel, the common band will be seen at 400 while the mutant will be seen on 200 base pairs. What if you have both the alleles? You will get three kinds of amplicons: one common amplicon, one for wild allele and one for mutant allele. When you run the sample on the gel, you will get three bands, which shows heterozygosity. Now let's switch the primers. In this case you will get two amplicons, one for wild and one common. Here you will get a 300 base pair amplicon for mutant and 400 base pair common amplicon. These can be visualized on the gel. And this is the last case. This one is again heterozygous case. But this time the 200 base pair amplicon will be for wild and 300 base pair for mutant and 400 base pair common. So that was all for today. Don't forget to subscribe my educational, entertainment, and animation channels. Thanks, bye.